Hello and welcome to another Saints Spotlight. Today's guest is Kevin Burridge, an alumnus from the class of 2016. Kevin is currently pursuing his doctorate at Miami University and is conducting cutting-edge research with his professors in bioconjugates. Thanks for joining us, Kevin.
Hi, great to be on.
Tell me, Kevin, when did you know you were interested in science?
I have been interested in science from a very young age. I was always a very curious kid. Like many boys, I loved dinosaurs, and I wanted to be a paleontologist and, dig up dinosaur bones. I read all kinds of fact books about animals and plants and bugs, and just discovery in general has always been exciting to me. I mean, I'm the guy who browses Wikipedia when I'm bored just because I like to learn how things work.
You mentioned when you were at Thomas More that you were very involved with your organic chemistry professor. What project did you do, and who was that?
I worked with Dr. Dan Esterline on a project basically trying to make a penicillin derivative because at the time I was interested in beta-lactam-resistant bacteria.
So what drew you to Miami University for your graduate studies?
My academic and research background was all like cell biology and molecular genetics. I didn't actually add a chemistry major until maybe my junior year. And so even though I had a good academic record, none of the top 10 PhD program schools that I applied to for chemistry were very interested in me. Dr. Esterline, though, is actually a Miami alumnus, so he suggested that I apply at Miami and that it might even be a better fit for me. When I visited Miami, I was really impressed with the campus. I was also impressed with the department here, which actually receives quite a bit of external funding and has some top-notch instrumentation. That includes the EPR facilities, which are actually the biggest EPR facilities in the Southwest Ohio. And then there's an instrument called NMR, which organic chemists use routinely, and they have a lot of those here as well.
Well, it sounds like it's been a great opportunity for you with your research. Can you tell us a little bit about the scientific project that you've been working on with the bioconjugates?
So to give it some background, proteins, and particularly enzymes, are the functional macromolecules that keep your body running. A protein molecule's function actually comes directly from its shape, which we call its structure. You can think of a protein kind of like a paper plane. So if it's folded properly, it'll perform well. The paper plane will fly, the protein will do its reaction. But when proteins or paper planes unfold, they stop working. And like a paper plane, proteins also can experience wear and tear from use, and they'll eventually just unfold and fall apart. Our little slice of bioconjugates is understanding how we can put synthetic polymers on proteins to make them more functional and stable. And the main project I'm working on is trying to unravel the mystery of how polymers stabilize proteins at a microscopic level. The field currently isn't even sure how each polymer behaves when it's attached to a protein. We don't know if there's specific interactions at the protein surface, or if polymers wrap tightly around the protein like mummy bandages, even if they spend time close to the protein surface at all. And depending on the protein polymer pair, any one of those things could be true. To be able to answer these questions, we adapted a method called Paramagnetic Relaxation, Enhancement, or PRE, our protein polymer conjugates. In short, we make these polymer chains of molecules that are linked together, and most of the chain is made of the polymer of interest that would be used in an actual, downstream application. But then we have these reporter units also incorporated into the chain at a predefined position along the length. Then we take those polymer chains and we stick them to the protein surface. Now you have a polymer chain with reporter groups attached to the protein. Now the reporters will actually cause change in the magnetic spectrum of the protein, and that spectrum directly relates to the protein structure. And so when the reporter is close to the protein, the spectrum will change significantly. And when it's further away, then not so much, or not at all. Using this method, we were actually able to find that the polymers do adhere weakly to the protein surface, and the polymers prefer to interact with a particular face of the protein. However, this work is just one data point. So we've got one polymer type and one protein. But we hope that can be adapted for other research groups as we continue to refine the method and use it for a wider variety of polymers and proteins.
That is fascinating research. What kind of everyday application would you use that for?
Proteins are pretty useful molecules. You want to stabilize them so that they can be useful over a longer period of time. There are examples in both medicine and industrial applications using proteins and protein polymer conjugates. There are many protein drugs now that have a polymer called polyethylene glycol attached to them. And that will actually significantly increase their circulation time after someone's been treated with it, which means that it'll be functional for a longer period. And that means people have to get fewer treatments, and the initial dose doesn't have to be quite as high.
Are there other projects that you're working at or thinking about tackling?
In my experience, good project ideas tend to come up much faster than they can actually be completed. I have another bioconjugates project where I am trying to efficiently attach two different polymer types on the same protein at defined locations, which is actually easier said than done at the microscopic level. But the idea of that is if you wanted a protein that, say, had one polymer that would contribute a lot of stabilization and then another polymer that would make it more easy to recycle, you could do both.
Okay.
So that would be useful for industrial and medicinal applications.
When you were at Thomas More, what type of student activities were you involved in?
I was a Chancellor Scholar, so that involved a lot of service and liturgical activities. And then on the science side of things, I have to give a shout out to Dr. Hageman because I took his cell biology class, and from one of the labs we went to the University of Cincinnati's microscopy lab. That's where we met the program director for UC's Summer Undergraduate Research Fellowship, and that kind of got my foot in the door, and I applied, and I was able to do 2 summers there as a paid intern doing molecular genetics research. I also did some independent chemistry research with Dr. Esterline in my senior year, and that was where I tried to synthesize a penicillin derivative.
Do you have any advice for current students?
My biggest piece of advice for junior and senior science majors would be to get research credit. That's for a few reasons. For me, I was able to use it to count towards laboratory credit. So that meant I got to do my own thing instead of doing more lab reports, which is great. You also get to interact with your professors more, and those are the people that are going to write you letters of recommendation when you apply for graduate school or jobs. That'd probably also look better on a CV. And finally, it's just good to get a feel for the actual grind of research, which can definitely be a roller coaster because, there are some months where nothing works at all and you feel like you're getting nowhere, and it's very frustrating. But, if you press on, you throw a whole book at whatever problem you have, eventually it usually works, or you'll find something, or you'll find a better way to do it anyway. So that's my advice.
Okay. Well, Kevin, thank you so much for being our Saint Spotlight.
Yeah, it's great to be on.